Good morning, all of you. Today we are going to discuss genes and regulatory elements. So, the elements which we consider as a regulatory, they are what the regulatory genes. Then there are structural genes are there, and regulatory elements are also there. Structural genes are the genes which encodes the proteins, while the regulatory genes encoding the products that interact with the other sequences and affect the transcription and translation of these sequences. Then the regulatory elements are there. The DNA sequences that are not transcribed but play a role in regulating other nucleotide sequences. They are called as what? Regulatory elements. Genes and regulatory elements, again we have to consider the constitutive expression, positive control, and the negative control also. Constitutive expression means the genes which are continuously expressed under the normal cellular conditions. Then these genes are positively controlled as well as the negatively controlled. The positive control stimulates the gene expression, while the negative control inhibits the gene expression. Now, there are the several levels of the gene regulation. Many levels at which gene expression may be regulated. They are the DNA access, then transcription, RNA modification and stability, translation, and post translational modification. So this slide indicates that how the regulatory elements and regulatory genes are present and how they are carry out their function. Now the second is what? DNA binding proteins. They contains the domain 60 to 90 amino acids which are responsible for the binding to the DNA, forming the hydrogen bonds with the DNA. Distinctive types of the DNA binding proteins based on the motif. So what is this motif? Motif is within the binding domain a simple structure that fits into the major grooves on the DNA. Then there are helix turn helix type of the motif are there. So here we consider the two helix and one is the turn. So this is the DNA binding helix, then which is in the form of the helix turn helix. Then zig fingers motif are there. So these are the fingers and these are the zinc ions are there. Then the leucine zipper. So this is the structure of that leucine zipper type of the motif. Now the operon which control the transcription in bacterial cells. So what do you mean by operon? Operons are nothing but the promoter and additional sequences that control the transcription, that is the operator and the structural genes. And the regulatory genes are the DNA sequences encoding the products that affect the operon function but are not part of the operon. They are the regulatory gene are there. So here we have to consider that what are the common types of the binding motifs are there. The motifs are helix turn helix, zinc finger, receptor are there, then leucine receptor are there, helix loop helix receptor are there, and homeodomain are also there. So this helix turn helix motif located in the bacterial regulatory proteins, related motifs in eukaryotic proteins, while the zinc finger motif are the eukaryotic regulatory and other proteins are also involved. In the steroid type of the receptor, eukaryotic proteins are there. Then the leucine zipper, eukaryotic transcription factors, and helix loop helix, they are the eukaryotic proteins. Then homeodomain are the eukaryotic regulatory protein. What are the characteristics of this helix turn helix? They are having the two alpha helices, and the zinc finger is nothing but the loop of amino acid with zinc at base. Steroid receptor to perpendicular alpha helical with zinc surrounded by 4 cysteine residues. Then the leucine zipper is the helix of leucine residue, and basic arm two leucine residues are ingredients. Then the helix loop helix type of the motif is there, which are two alpha helices separated by the loop of amino acids.